Hi everyone! Today I'm going to explain how viruses work. Viruses are fascinating because they're not technically alive. They don't have cells and can't survive on their own. Yet, they share many characteristics with living things, like having genetic material, reproducing, and evolving over time. Let's explore how viruses invade cells and replicate, starting with what makes them so unique. First, let's talk about what all viruses have in common. Every virus is made of a protective shell called a capsid, which is composed of proteins. This capsid encases the virus's genetic material, which can be either DNA or RNA. Think of the capsid as the armor that protects the virus's instructions for replication. All viruses have protein markers or spikes on their surface. These markers are crucial because they act like keys that bind to specific receptors on the surface of host cells, which act like locks. This matching process determines which cells a virus can infect. When the protein markers on the virus match the receptors on the cell, the virus attaches to the cell, like a key fitting perfectly into a lock. Inside the cell, the virus's genetic material takes control. Think of the cell as a factory and the virus as a hacker. The viral DNA or RNA reprograms the cell to stop its normal activities and instead start producing viral components like capsid proteins, enzymes, and new copies of the virus's genetic material. There are two main ways viruses replicate: lytic and lysogenic. Lytic viruses work fast. They replicate quickly and cause the infected cell to burst or lyse, releasing new viruses to infect more cells. Lysogenic viruses integrate their genetic material into the host cell's DNA and lie dormant. As the host cell divides, it unknowingly copies the viral DNA along with its own. When the virus is triggered by stress, a weakened immune system, or other factors, it activates and switches to the lytic phase, ultimately destroying the cell. But not all viruses destroy cells right away. Some, like the flu virus, exit the cell through a process called budding. Instead of bursting the cell, these viruses pinch off a piece of the cell's membrane to form their envelope, leaving the host cell mostly intact. This allows the virus to spread while the cell continues functioning, at least for a while. By studying viruses, we not only learn how to combat infections, but also how to use their unique properties for medical advancements. Thank you for watching.